All right, welcome back. Here we are. We just got done with our conversation on mutations and mutagens. So now what we're going to do is we're going to spend a couple slides on how we'd be able to identify mutants, bacteria that contain these mutations and are expressed in their phenotypes. So there's a couple different ways we're going to be doing that. We have positive selection, negative or indirect selection, and what's called the Ames test. So let's get going. Okay, so here's our first method. It's called positive selection of mutants. So we're just going to walk through this experiment, and in that process, it will explain kind of what's going on. So in this first test tube here at the top of the picture, you have a culture of bacteria, just a normal culture. It's not modified in any way. And in even normal cultures, there's going to be a likelihood that you might have a few bacteria that already contain a mutation that is just a randomly occurring mutation. So what we're going to be looking for in this particular test, we're going to be looking to see what percentage of those bacteria are resistant to penicillin, and if that number of resistant mutants increases if we add a mutagen to the mix. So let's do our control first. So the control is going to be this first half, part A. So let's say you have a bacterial culture. A few of them are going to be resistant, which means they grow in the presence of penicillin, and quite a few of them will be sensitive in that they die in the presence of penicillin. So we set up our two plates. We have our control plate where it's just a regular agar without penicillin. We plate everybody out. Everybody's going to grow. So penicillin resistant and penicillin sensitive, because there's no penicillin in this plate. It's just a normal kind of growth medium. In the left plate, this is a medium with penicillin. So it's going to kill off all of the ones that have the mutation, or sorry, that don't have the mutation. And the one with the mutation can grow because it's a penicillin resistant cell, right? So we got one colony grew out of this original culture that was just a random mutation. So then what we're going to do in our experimental part of this activity is we are going to add a mutagen. Maybe something you're testing is this particular substance, this chemical or exposure to light or some condition, does that increase mutations in bacteria? So then what you'll do is you'll get your tube, you'll add the mutagen that you're hoping will increase mutations. You add that in, you plate it out again with penicillin and without penicillin and see what you get. So everybody's going to grow on the plate without, because that's just our control plate. And then taking a look at the one that has the penicillin of the bacteria that are exposed to the mutagen, we have 5 colonies. And so what does that mean? Well, it makes it clear that the mutagen, whatever you added to your test tube, induces cells to go through mutation, and they just happen to mutate the gene that allows them to be resistant to penicillin. There's probably mutations elsewhere, but that's not the test we're running. And so you can do a little calculation to figure out the mutation rate of that particular mutagen. So basically what you do is you take your 5, right? So that's the 5 here, mutants that came after you adding the mutagen, subtract the 1 that came from just your normal wild type, just random mutation, divide by the 1, right? So 5 minus 1 is 4, divide by 1 is 4, times 100 gives you a 400% mutation rate. So whatever that mutagen was, this guy right here, it increased mutations by 400%. Okay? If you're to do that test, you had 1 random mutation, you added your mutagen, and you had 1 mutation, it would be 1 minus 1 divided by 1 is 1. Or 0, it would be 0 mutation rate. So you wouldn't have any increase in mutations after adding your mutagen. So it's probably not a very convincing mutagen. It maybe doesn't mutate DNA. And so this is ways that researchers can use bacteria to test for the ability of particular substances or conditions to induce mutations. And the reason why this is called a positive selection is we can positively see it with our eyeballs. The way that this mutation is set up is this resistant mutant grows on the presence of a plate with penicillin. So just the visible growth of those mutants is the positive selection part. Now that might make a little bit more sense once we compare that to the negative or indirect selection, which is next. Right? So in this case, we're not going to be looking for mutants that might grow on a penicillin plate. In this case, we're going to be looking for mutants that are what we call auxotrophs. Okay, an auxotroph is a bacteria that's lost the ability to make a growth factor. So in this case, we're going to be looking for tryptophan auxotroph. So we just got done going over the trip operon, right? So we know that the bacteria have the trip operon. When it's expressed, they can make their own tryptophan to be able to incorporate it into their proteins. Well, a tryptophan auxotroph would have lost its tryptophan operon. Either maybe the promoter got mutated, or one of the genes got mutated, or the operator got mutated, or something, some part of the trip operon is mutated. And so the cells cannot produce their own tryptophan. And that's what we call an auxotroph. They've lost the ability to make their own growth factor. So what we're going to do is we are going to take our bacteria, just a normal kind of a culture. We're going to add our mutagen, right? So that's our whatever we think it is going to be that might trigger this mutation. And then we are going to plate it out on a petri dish that contains tryptophan. So even the auxotrophs should be able to grow, the ones that can't make their own because they're getting it for free from the environment. So then you get your growth, right? So you get your plate, but you can't tell which of those bacteria could be mutated auxotrophs because everybody's growing on the tryptophan plate. So what you need to do is you need to grow them on a plate that lacks tryptophan, because then your tryptophan auxotrophs won't grow. But the problem with that is, how would you know? So if you plated out all of your mutagen exposed bacteria on a plate that didn't have tryptophan at the very beginning, the only ones that are going to grow are the ones that can make their own tryptophan. The mutated auxotrophs won't grow, and you wouldn't know any different. You're like, oh, this mutagen doesn't do anything. So what you need to do is you need to set up your the growth of all of the bacteria, and compare that to a plate that lacks tryptophan. And the way they do it is a very cool process. It's called replica plating, right here. So you take your plate that had everybody growing on it on the tryptophan medium. You take a sterile piece of velvet. You put it on this big rubber stump, like a stamp pad. You line up the little X's right here and right here. You line those up, and then you stamp down onto your plate that had all of the bacteria growing on it, and you stamp that onto a plate with tryptophan, and you stamp that onto the plate without tryptophan. So the reason why we use velvet is because the velvet has those little hairs on it, and it picks up bacteria from those colonies, those first original colonies. And then when it touches the sterile surface on the media, it transfers some of those cells. So what you end up getting is everybody's growing on the plate with tryptophan, and on the plate without it, the auxotrophs won't grow. But because you've done an identical inoculation with this replica plating, you can find which colonies on the tryptophan plate are the ones that can't grow without tryptophan. And so it would be this little colony right here, because it's missing right here. And so then you can take your loop and you can pluck that little colony off and put that into a test tube, and now you'll have your 100% pure culture of your tryptophan auxotroph. So the black and white picture here is showing some of those, an actual outcome of a replica plating. So I'll just highlight, right? So this colony is here, this colony is here. These two colonies here are missing in that particular location of the petri dish. So these guys with little black dots are the ones that are the auxotrophs that grew on the plate with the nutrient. And the mutants don't grow on the plate without the nutrient. All right, so this is called indirect selection, because you need to have everybody grow to be able to grab the thing that is the mutant that didn't grow on the plate that is your thing that tells you that that's the mutant. I don't know if I explained that very well. Send questions or come to office hour. Let me know if that was a little bit confusing. All right, our last conversation in our identifying mutants is with identifying carcinogens, actually. It's a little bit more human focused than just looking for bacteria. This is called the Ames Test. And so the Ames Test is a way for researchers to do a quick and easy screening of certain chemicals to identify if they could be potential carcinogens. Because running these tests on humans would not be ethically correct, and they'd probably be really expensive. And so we want to see if we can at least get some of the things that are safe for bacteria are most likely going to be safe for humans and not even spend money or research time on those. So this particular test uses a auxotroph called His-negative Salmonella. So what that means, it's a strain of Salmonella that is an auxotroph that cannot make its own histidine. So it's lost that operon, or the ability to make its own histidine. So the experiment goes, let's have a suspected mutagen. This guy here, does it cause DNA mutation? We don't know. Let's test it. So they put the suspected mutagen in your culture of His-negative Salmonella, and you'll also see some liver extract is going into the tube. Now, the reason why liver extract goes into the tube is because a lot of times the things that you consume are processed in the liver with lots of enzymatic pathways. So if you didn't have the liver extract, you wouldn't get a more realistic view of that potential mutagen of how it would actually react in a human body, because we all have livers. And so sometimes something that you take in is not carcinogenic, but after it gets processed by the liver, it turns into a carcinogenic derivative. And so we add those liver extracts to mimic the functionality of the liver. So in your experimental tube, you have your His minus bacteria, your suspected mutagen, and your liver extract. In your control tube, you have the His minus Salmonella, your liver extract, but no mutagen. Okay, and then we plate them out on a plate that does not have histidine. Right, so these are His-negative Salmonella. If we plate them out on a plate that does not have histidine, we should expect no growth, and that's exactly what you get in the control tube. In the experimental tube, if your suspected mutagen actually mutated DNA, and if that mutation affected that particular operon and made the mutation so the Salmonella can make their own histidine, it's called a revertant. So whatever that mutation was, it reverted it from a His-negative to a His-positive, and now the bacteria can make its own histidine, so you get some colonies that are growing on the presence of that histidine-lacking medium. That's a big process. So what's the point? Well, the point is, if you have the suspected mutagen that produces revertants, these mutated Salmonella that can produce their own histidine, that is a mutagen that can mutate DNA. So it might warrant further research. If it doesn't, if you put the suspected mutagen and there's no back mutation, then let's not do any further research on that. Because remember, this is looking for carcinogens. Carcinogens are things that cause cancer. Bacteria don't have cancer. Cancer is a disease of multicellular organisms. So this Ames test is specifically for looking for substances that can have a negative impact on multicellular organisms, primarily humans, right? Because this is who we want to spend most of our money on, is making sure that we are not exposing ourselves to substances that could potentially mutate our DNA that could lead to cancer. So the Ames test is a really quick and cheap and easy way to screen lots of different kinds of substances in maybe a larger scale safety test for a particular chemical or substance. Okay, all right, so we did positive selection, negative selection, and Ames test for mechanisms of identifying mutants. All right, see you next time.